I'm Bruce Ames, and I'm a senior scientist at Children's Hospital of Oakland Research Institute and an emeritus professor at UC Berkeley. I'd like to tell you why I think when you're short of any of the 30 or so essential vitamins and minerals, it ages you faster in some way, and that this is relevant to most of the population of the world. So this is your metabolism. What do you need to run your metabolism? Well, you clearly need fuel, fat and carbohydrate, and oxygen, and you burn your fuel in the mitochondria to make kilos of ATP every day. And that powers your muscles and your brain and everything else, all the uphill metabolism. And in addition, you need about 15 or so minerals and 15 or so vitamins, and these are cofactors for proteins that do some of your metabolism. If you don't get any one of these 30 substances, you die. But how much do we need? I think people really don't have a clue, because they're all thinking short term, and I think we should be thinking long term, and I'll explain why. So, are we getting enough of these vitamins and minerals? Well, the committee set an RDA and an EAR, a measure of inadequacy. But again, it's based on short term, not long term, and I'll tell you why that's important. So these are roughly the 40 essential micronutrients that we need. They're roughly 15 minerals and 15 vitamins, and then 2 essential fatty acids, the omega-3s and omega-6s, and some essential amino acids and choline. Anyway, there are 40 total substances, but mainly today I'll talk about the vitamins and minerals. So how much do we need? Well, the committee sets up 2 numbers, the EAR and the RDA. The EAR is some distribution in the population, and if you're below the EAR, the committee says you're inadequate. And then they set the RDA at 2 standard deviations above the EAR. So they're really linked numbers. And just taking the current numbers, who's below the EAR, well, in iron, it's 16% of menstruating women. Women are losing iron all the time when they're menstruating. 16% of them are not getting enough by the current standards. Magnesium, we're talking about 56% of the whole US population. Where do you get your magnesium? Well, you get it from anything that's green, because it's in the center of the chlorophyll molecule. So you eat a big plate of spinach, you get your magnesium. You don't, you don't. Now, you can also get some from almonds and other nuts, but most of what we get in our diet is from greens. Now, magnesium's in 400 or 500 enzymes, practically every DNA repair enzyme and all sorts of other important enzymes. And when you don't get enough, you're disabling lots of them. Zinc is in 2,000 proteins with zinc fingers. So it's turning on all the enzymes that recognize sequence in DNA. Most of them have zinc fingers. And it's in copper zinc superoxide dismutase. And it's P53, which is involved with cancer. So every micronutrient is important in some way. And 12% is below the EAR. B6, half of elderly women. Folate used to be huge numbers, and I'll talk about that. Now we fortify the flour, and we still have 16% of adult women are low. E, practically everybody. C, a third of people. And then we're very low in vitamin K, vitamin D, calcium, potassium, omega-3. What got me interested in this field, I was interested in oxidants being made in your own body as byproducts of your mitochondrial metabolism, and then I got interested in antioxidants, and we turned up a few that people hadn't thought about before. But what really got me into this field was when Jim McGregor came to my lab on sabbatical. And Jim McGregor was a cytogeneticist, and he had been looking at chromosome breaks from radiation and what influenced it. And people had developed a neat new method. You can take red blood cells, which you see here, put them out on a microscope slide, and look for pieces of DNA in them. Now, you all know red blood cells don't have any DNA. When they make the red blood cell, the precursor cell has a nucleus that gets extruded, and then you make the red blood cell. But if there's a chromosome break, that little piece of DNA without a centromere gets left behind, and you can stain with a fluorescent reagent for DNA and find it. So if you look at a mouse, 1 in 2,000 red blood cells has a piece of DNA in it. You irradiate the mice, you get more. Well, McGregor stumbled on the fact that folic acid deficiency does the same thing as radiation. And so the more folic acid deficient, the more chromosome breaks. So it was just like irradiating the mice. And that intrigued him, and he wanted to do it in people. But this method doesn't work in people because the function of the human spleen is all the blood goes through the spleen, and any cell that's a little stiff gets filtered out. And inbred mice don't have any splenic function. That somehow got lost in all this inbreeding. So this doesn't happen in mice, and you can measure this very nicely. But in people, it doesn't work. So McGregor went to Kaiser, our local health maintenance organization, and found 20 people with no splenic function. You can lose a spleen in an accident, and Kaiser has hundreds of them in their 6 million person database, and they let McGregor contact these people, and the people lived perfectly fine, and see if he can get some blood. So he got some blood, and the baseline here is 19 of those 20 people had a baseline of a few broken chromosomes per 1,000 red blood cells in humans. But there was one person of the 20 he looked at who had maybe 25 times the level of everybody else. So McGregor thought that was very interesting, and he followed this person for a year. He's following both the reticulocytes, which are the newly synthesized red blood cells that normally don't have any DNA, but you can identify them by a marker on the surface, and then the regular red blood cells. So when he discovered that folic acid deficiency breaks chromosomes in mice, he thought, well, he'll go look at this guy, was he low in folic acid? And in fact, he was low, but not wildly low. And so he gave him folic acid for a couple of weeks, and right away the retics came right down to baseline. And the red blood cells take longer to turn over, and they followed. So what's beautiful about this is you see causality right there. And then they ran out of the folic acid, and they started going up again. Again, the retics go up faster than the red blood cells, which you see immediately. And then McGregor thought it would be unethical to do any more experiments on this fellow, so he said, well, either you have to eat a better diet, or I'll give you a folic acid pill. And the guy said, well, give me a folic acid pill. And so he gave him a folic acid pill, and both the retics came right down within a few days to baseline again, and the red blood cells followed. So what's nice about this is he can see the exact level of serum folate or red blood cell folate, which is 20 or 30 times higher, and measure that and see what level causes this. So not only do you get causality, but you see what level does it. And that level that was this fellow had was in half the poor when you look up the numbers, and about 10% of the US population. So it's a huge thing. I had been spending 5 or 10% of my time debunking environmental fears about parts per billion of pesticides. All of that just didn't sound plausible. That was anything that we wanted to worry about. It. The levels were too small. And here's something that was a huge risk. And diet, Americans are eating a terrible diet because we're eating all this refined food and we're not getting our vitamins and minerals. And so anyway, this got me all activated. And we worked with McGregor to figure out the mechanism, which we did. So what you can see here is Fenwick at about the same time as our lab published a paper comparing X-rays to folic acid, Fenwick in Australia. and I'll show you his slide. So the more X-rays you get, the more chromosome breaks you get, and the less folic acid you get, the more chromosome breaks you get. We both did similar studies. Half the poor are breaking their chromosomes because of folate deficiency. It's well pinned down that broken chromosomes lead to cancer. And people worry about incredibly tiny levels of radiation from Fukushima or from other tiny sources of radiation, but we're not worried about our diet. So we helped McGregor work out the mechanism. There are two main pools of folate: methylene tetrahydrofolate, which was discovered at Berkeley many years ago, and methyl tetrahydrofolate. Well, methylene tetrahydrofolate converts DUMP to DTMP. It puts that methyl tag and doesn't affect the base pairing, but T is in DNA and U is in RNA. And methyl tetrahydrofolate in B12 converts homocysteine to methionine. And methionine then goes to S-adenosylmethionine, which is the main methylating agent in the cell. And that then regenerates homocysteine. So you're always making more homocysteine, and people have shown when homocysteine accumulates, that's a risk factor for heart disease, and it's a risk factor for cognitive dysfunction. And so the evidence on cognitive function is really pretty strong, and the evidence on heart disease had been a little controversial, but particularly it involves particular polymorphisms in the population. And methylene tetrahydrofolate reductase converts DUMP to DTMP. So 4 or 5 types of cancer have been associated with folate deficiency. So if you don't convert uracil to thymine, you're putting uracil in your DNA, and that's always being repaired out and causing nicks and breaks. And if you don't, homocysteine accumulates, that's associated, as I say, with heart disease and cognitive dysfunction, and that's due to homocysteine accumulating, and people sort of understand the mechanism. And this polymorphism, so Northern Europeans, 40% of them increase the lower pool at the expense of the upper pool. So they get less cancer and more heart disease and cognitive dysfunction. Now, why is that? Those come late in life, and you wouldn't think that would be a strong element for selection. So we suspected it was sperm, which nature really cares about. And in fact, you can show that when you're low in folate, it affects your sperm. Lynn Wallach in my lab had a paper on that. So in any case, I think many of the polymorphism people are discovering are going to have to do with diet. And it's clear that when you muck up your metabolism, you can have all sorts of things going wrong. And I'd like to give you the mechanism of that. So why does incorporating uracil in DNA from not having enough methylene tetrahydrofolate cause chromosome breaks? Well, the radiation biologists have worked out the mechanism from radiation, and the most dangerous part of radiation is when you get 2 nearby lesions, one on each strand. So, of course, the strands are around each other. And then repair enzymes are always cruising along the DNA looking for an oxidized base, which you get from radiation, or looking for a uracil. And they clip that base out, the other strand holds it in, and now you repair it. But if there are 2 nearby lesions, then if within a short time you take out both lesions, then the chromosome falls apart. And that's double-strand break is considered the most dangerous lesion from radiation, and you're getting the same thing by the time you put hundreds of thousands of uracils on the newly synthesized strand, and you have 70,000 or so lesions from oxidation on the other strand. So it's just like radiation, really. So after looking at folate and helping McGregor work out the mechanism, then the next micronutrient I got interested in was iron. Fernando Viteri, a professor in the nutrition department, has been interested that there are 2 billion women and children in the world who are getting too little iron. And it affects the brain. There's really very solid evidence on that in mice and rats and kids. And it was known that too little iron damages mitochondria. And we were experts in mitochondria, and vitK came to us and asked to collaborate on this. So we set up assays for all kinds of different mitochondrial functions and looked at the whole range of iron in mice. And too little iron is up is bad on this plot. So up means the mitochondria are pouring out more mutagenic oxidants, they're getting less efficiency, they're getting more mitochondrial DNA damage from oxidation. And then there's a window of iron, which is the ideal amount, and then too much is bad for you. But too little is 2 billion women and children in the world, and too much is a lot of old men eating too much red meat. And minerals are tricky because vitamins you can usually get lots more than the RDA and it won't hurt you. Vitamins are relatively, the body's tolerant. But minerals, one mineral looks like another. So iron looks a little like copper and zinc. So if you have too much iron, people think it may be interfering with copper uptake or interfering with zinc uptake. And then you have other problems. So in any case, it's clear that too little or too much wrecks your mitochondria. So, and then Emily Ho came to the lab and she was interested in zinc. And so she grew human cells in culture and made them a little zinc deficient. And you get many more oxidants being poured out by the cells, presumably out of the mitochondria. And you got more oxidative damage from these mutagens that you're producing. And then Hania Tam in the lab looked at biotin, and if you make human cells in culture a little biotin deficient, they fill up with oxidants. And then we looked at mechanism, and we came up with a plausible mechanism to do with heme biosynthesis in the mitochondria. I won't go into the details there, but one can track down why each of these causes problems. But basically, you're getting the mitochondria to pour out more oxidants, which happens with aging in any case, and less efficiency. So it's like an old car engine, an old car. They pour out more black smoke, more side products, and the side products in this case are oxidants, and less efficiency. So basically, in your mitochondria, you're burning fat and carbohydrate, which means adding 4 electrons to oxygen to release the energy. And if you add the electrons one at a time, you get superoxide and hydrogen peroxide and hydroxyl radical. And that happens with aging normally, but it happens when you're short of vitamins and minerals. And David Killian in the lab showed that magnesium deficiency induces mitochondrial DNA protein crosslinks. So you're getting radicals and cross-linking with protein. It may be other shortages do the same thing, but in any case, he could show it with magnesium deficiency. So then we plugged into Google 30 different vitamins and minerals and put it against mutation, cancer, chromosome breaks, and all this literature popped up. And again, it's epidemiology, so you can't always, some of the literature is fuzzy, but calcium deficiency has been associated with chromosome breaks and colon cancer, folate deficiency. We did some experiments, McGregor did some experiments, Fennick did some experiments, chromosome breaks and cancer. Vitamin D deficiency has been associated with cancer, magnesium deficiency, zinc deficiency, potassium deficiency, choline deficiency, niacin deficiency, omega-3 deficiency, selenium deficiency, vitamin B12 deficiency, and who knows how many others. This is just what people have looked at. Now, one of the reasons nutrition is such a messy science is we're relying for causality on epidemiology, which can't really show causality. As you all know, epidemiology is fiendishly difficult. It doesn't mean we shouldn't use it, but half the time people get the wrong answer. The epidemiologist joke is Miami's a weird place. Everybody's born Hispanic and dies Jewish. That's called confounding. And you have to be awfully good and understand mechanism and understand what's plausible. And all you're getting is an association, which could be wrong, because healthy people do lots of healthy things and unhealthy people do lots of unhealthy things. And we're mechanism people, so we're trying to work from mechanism on up. So about 6 years ago, I kept on thinking about all of this. Why is nature giving you cancer when you're short of vitamins and minerals. And then it occurred to me one day that that's what nature wants. It's a trade-off between short-term and long-term. So the vitamins and minerals aren't spread evenly through the world. The minerals aren't spread evenly on the earth. So there are areas with high selenium and low selenium and high iron, low iron. The vitamins aren't spread evenly through plants. So animals have always been running out of vitamins and minerals. And what we postulated is that they built into a metabolism this trade-off between short-term and long-term. When you're running out of magnesium or vitamin K or whatever, the body keeps those proteins that need that vitamin or mineral for survival and reproduction. Those proteins keep it, and the ones that are more long-term, that preventing your dying sooner from disease of aging, those lose it. And I call this triage, and that means most of the world's population has an inadequate intake, which we're not eating the perfect balanced diet. Anyway, the trade-off between short-term and long-term has been discussed relative to aging by evolutionary biologists, but people hadn't really thought about it in terms of the biochemistry. And now I think we've shown it's really true. So Joyce McCann in my lab, who likes to do theoretical work and is very smart, came to me one day and said, I'm a little skeptical of your triage theory. I think there's a better way of tackling it. And I said, Joyce, go to it. And so she looked into which vitamins and minerals were well studied but not too complicated. Vitamin D is way too complicated. It's a hormone. It goes to a hormone that's turning on 900 genes. Zinc is in 2,000 different proteins. Magnesium is in 400 or 500 different proteins. But she picked some that were not too complicated but well studied, and she started with vitamin K and then did selenium, and they're both beautiful because this is built into metabolism, this trade-off between short-term and long-term. So I'll go through vitamin K because it's so interesting. So vitamin K, K stands for coagulation in Dutch, because a guy named Dam many years ago showed that there was a compound in plants that animals used for making blood coagulation proteins. And so what Joyce did is look at the mouse knockouts, which ones are essential for life and which ones you can do without. And the coagulation proteins are all essential. You knock them out in mice and they're all embryonic lethals. So we're not interested in those. Those are the short-term survival ones. But we're interested in the ones where when you knock it out in mice, you get some disease of aging, and those are the rest. So osteocalcin's in the bone, it moves all these proteins are calcium binding proteins, because what vitamin K does, it's used in photosynthesis in plants, a compound called quinone, and animals use it as a cofactor to take a glutamic acid, an already made protein, and put another carboxyl group up at the top. So that makes a compound that can bind calcium, and they all bind calcium. So osteocalcin's in the bone, moves calcium around in the bone. You knock it out in mice, and the pups are born, they look perfectly okay, but you stress them a bit and the bones break. So it doesn't make a strong bone if you don't have that protein. And one of the risk factors for having vitamin K deficiency is bone fractures. And people who take Coumadin or warfarin, 30 million prescriptions a year, which interfere with your vitamin K metabolism, those people also get many more bone fractures. So, and GAS6 is an interesting one that has to do with acute coronary syndrome, and it has to do with diabetes. And matrix LA protein is very interesting because that protein, when you knock it out in mice, the pups are born, and then they all die at about 3 months of age of calcification of the arteries. Turns out the function of that protein is whenever hydroxyapatite crystallizes out in your blood vessels, which is a calcium phosphate crystal, this protein grabs it and takes it out of there. So it prevents calcification of the arteries, which is a major risk factor for heart disease. And lo and behold, the people who get Coumadin, warfarin, die at 9 times the rate of calcification of the arteries, and it's a risk factor for low vitamin K. So everything kind of fits. If you look at what happens to the mouse gene, you look at the human gene as a rare genetic mutation where people died at an early age of calcification of the arteries in the corresponding human gene. And then TGFBI is cancer. The animals all die of cancer. And one understands the mechanism. This protein's in the matrix. It's interacting with integrin on the surface, which interacts with the microtubules. And if you don't have that protein, mitosis doesn't work as accurately. So you lose a chromosome here or there. And that's a risk factor for cancer. And that's exactly what these people get. Okay, so this shows osteocalcin matrix R protein. And a very good Dutch group has worked on this, and there's a very good group in Boston, Sarah Booth, and Schurgers and Vermeer in Holland. And we didn't do any of the original work. We're just coming in as theoreticians. But what they showed is the coagulation proteins are all done in the liver and get first crack at the vitamin K. When you eat anything green, you're getting vitamin K because it's used in photosynthetic plants, goes to the liver and does those proteins. And then the K is converted to MK, menaquinone form of vitamin K, and that has the property of going to all the tissues equally, and all these other proteins are done in the non-hepatic tissue. So right away you see why the coagulation proteins get first crack at the vitamin K. They sell MK7 in the health food stores. You could give MK7, and that way you'd be doing all the tissues equally and helping these other proteins that are being disabled when you're a little short of vitamin K, which is probably half the country. This is a Japanese health food, natto. Some of you may have eaten it. You get it for breakfast in Japan. It's a Bacillus subtilis fermented soybean. You put your spoon in, you get these long, white, yucky strings. You smell it. It smells a little yucky. You taste it. It tastes a little yucky. So Westerners don't like it, but the Japanese consider it a health food, and lots of Japanese eat it. Well, the people who eat natto, and it's filled with a compound called MK7. So this form of vitamin K that gets to all the tissues equally. And these people don't get very little heart disease, very little prostate cancer, very little bone fractures. So the epi all kinds of fits with this mechanism. And so it means right away we have people coming to the doctors with calcification of the arteries. There's something you can do about it without disabling all your blood clotting, which is important and is essential for life. So it's long-term versus short-term. Joyce McCann also looked at selenium, and again, the workers in the field had shown what happens with mouse knockouts, what's the hierarchy. There's both a tissue hierarchy and there's an enzyme hierarchy. And again, it's all long-term: cancer, DNA damage, heart disease, reduced resistance to infection, your immune system, poor cognitive function. So it's this trade-off between short-term and long-term again. And the proteins that are essential for survival and reproduction hang on to it, and the other ones lose it when there's shortage. And again, we're talking about lots of people in Europe are low in selenium because selenium is very patchy in Europe. But in the US, selenium's high in the states where they grow a lot of grain, and we ship food all around. So most of us are probably pretty good on selenium, but not all of us. And the mechanism that selenium uses is completely different from the mechanism that vitamin K uses. And it's quite interesting because it's a modified base in a transRNA. There's a special selenium transRNA that does only the essential proteins, and it all has to do with a methyl modification, which is inhibited by selenium deficiency. And we had done some work many years ago about modified bases in tRNA and regulation. So I think that's what modified bases in tRNA are being used for. There are lots of modified bases, but that's another whole story, which I won't go into. So then your immune system is being trashed when you don't have a good diet. So we looked up in Google, Swapna Shenvi, a postdoc in my lab who's working on this, we looked up in Google what goes wrong with adaptive immunity with aging. and these 6 things go wrong. And then we put in all the vitamins and minerals. Lo and behold, there was a fair amount of work on it. And so somebody looked at vitamin A and it changed CD4-CD8 ratio. Somebody looked at zinc. And then that folate paper was one of my own papers that I'd completely forgotten about. I had a French-Canadian postdoc, Chantal Courtemanche, who showed that folate deficiency changed the CD4 to CD8 ratios in T lymphocytes. We didn't understand what it was about, but we published it in Journal of Immunology just as an odd observation. But it's part of this triage, the trade-off between short-term and long-term. So to conserve the folate, when you're short, you trash your adaptive immunity. But that saves you from dying now. So it all makes sense, and it's not only cancer and bone fractures, it's every disease, I think, that we associate with aging. There's some insidious damage going on. You get DNA damage, it shows up as cancer in 10 or 15 years, but if it's a matter of survival, you trash that metabolism. And so most of you are walking around with not optimum metabolism, and it's aging you fast. So Joyce McCann, who did this wonderful work on the analyzing literature, had 4 big reviews about vitamins and minerals and the developing brain. So when you're developing your brain, you're making 1 trillion neurons. Each neuron has something like 100 to 1,000 connections. And if you don't get your iron, the rat pups don't do well on rat IQ tests, the mouse pups don't do well on mouse IQ tests, and kids don't do well in school. Well, vitamin D is a really interesting one because it's all involved with skin color. A dark skin protects you from getting disabled in the tropics because of ultraviolet light damage. And we have racially completely different people, Africans, Southern Indians, people in New Guinea. You select for a dark skin in the tropics. But if you go up north, where they're getting very little sunshine in Sweden or in Ireland, you're selecting for a lightest skin as possible. So to get that UV to make your vitamin D, you convert 7-dehydrocholesterol in your skin to vitamin D precursor, which then goes to make calcitriol, the hormone, and it's turning on 900 genes. Vitamin D receptor is in genes in the brain all over. So you look at the neurons, they have the enzyme that activates vitamin D to the hormone. You look at all the enzymes in the brain, and calcitriol is turning on all these different proteins. So you look at the brain, you can see the sequence where the vitamin D receptor interacts with the gene. And you have all these genes in the brain that calcitriol, the vitamin D hormone, is turning on. There are 900 of them. And so your brain doesn't get done well if you don't have vitamin D. And we have 95% of African Americans are vitamin D deficient. Now, maybe there are other differences and it's not a problem, but I think we should really look into all of that because it's something that's cheap and you can do something about it. People tend to think if it's genetic, you can't do anything about it, so therefore it can't be genetic. But in fact, if you understand it, you can often do something about it, and vitamin D doesn't cost much. So you put an Irishman in Australia, they're in deep trouble, and the solution is a hat and sunscreen. You put an African American in Chicago, they're in deep trouble, and the solution may be a vitamin D pill that costs a penny or two. So this is a slide of Fenwick, who looked at the variability in healthy, non-smoking males and females. And look at that variability. This is in chromosome breaks. There's a huge variability. What's it due to? I'll bet it's due to diet, a lot of it. And men have more damage than women, but women live longer than males. So we know that radiation shortens your life and does all these nasty things. Well, you don't eat a good diet, it's like irradiating yourself. So this triage analysis is really very beneficial. One, it provides a mechanism for how moderate vitamin mineral deficiency increases risk of a disease of aging and suggests a prevention strategy because we can either measure a particular protein or we can measure the DNA damage or whatever. And it indicates about half the proteins analyzed are longevity proteins. That's from Joyce McCann's analysis, which means that there's going to be a whole class of vitamins and minerals that nobody's discovered, because all the vitamins and minerals have been discovered on the basis of short-term effects. And we think, in fact, that we've discovered one. Jung Soo in my lab has been working on a compound called ergothioneine, which will interest you people because it's in mushrooms. And ergothioneine, he understands what it's doing in biochemistry. And it's something protecting you against a particular type of oxidative damage. Yet the transport systems that pump it in, we don't make it, but we pump it in when we eat a mushroom or a moldy cheese. And that transport system has been knocked out in mice by Japanese, and the animals live. Whoop! So it's not a vitamin by definition. But that's wrong, because that's the wrong way to think about it. Because a vitamin's there are these longevity vitamins that are going to keep you in a long lifespan. Anyway, that's another whole lecture that I don't have time to give. So obesity is a complete disaster. It's every possible disease is linked for obesity, years of huge health costs due to metabolic syndrome and cancer and heart disease, you name it. And cognition too, your brain goes out. The conventional wisdom is obesity is calories in and exercise out. But I think it's more than that, because obese people are eating the worst diet in the country. They're starving for vitamins and minerals because they're eating all this refined food and empty carbohydrate. And I think when nature makes you short of calcium or short of magnesium, the body is craving for this missing vitamin or mineral, and it makes you hungry. So hunger may not be only calories, it may be vitamins and minerals as well. And that would make sense. And it's been shown that obese people are hungry all the time, even though they're getting all these calories. But I think it's because they're starving for vitamins and minerals. Anyway, there's one paper that suggests that's true for calcium, and I think people have to work on that more. So these are the top sources of nutrition in the United States from NHANES. The main source of calories in the United States is sugary soft drinks, 9% of calories. And then pizza, beer, hamburgers, white bread, cake, donuts, pastry, French fries, fried potatoes, potato chips, corn chips, popcorn, rice, cheese or cheese spread. It's all refined food, and there's nothing green in there and no vitamins and minerals. So you have to eat way more veggies, and it's what your mom told you. So Mark Shiganaga, my colleague, and I have come up with a way to get vitamins and minerals into the poor. People have been telling the poor to eat the 9 portions of fruits and vegetables for years with zero success. But we think this may help in the problem. You could tell them to take 10 different pills, fish oil pills and calcium magnesium pills and all these things, but we put it all in a bar along with fiber. Fiber is very important for gut health, according to Mark Shiganaga. When you don't have your fiber to slow things down, and you get a bolus of sugar, you poke holes in your gut, bacteria get through, and you get all inflamed, and that's what's driving obesity. Anyway, there's starting to be a fair amount of evidence, but that's all my colleague Mark has figured out. So anyway, we made a bar, and we've done about 11 clinical trials, mostly on healthy people eating good diets. And it does wonderful things. It raises everybody's HDL. HDL is the good cholesterol. And as you can see, no matter where you started, most people, the beginning of the arrow is where they started, and 2 weeks after eating the bar is the top of the arrow. So we've done all these clinical trials. We can lower LDL, raise HDL, lower your homocysteine, lower your homo-IR, lower your blood pressure. Anyway, we have lots of good things going on in normal people on healthy diets, and we're starting to expand this to obese people. So should you just take pills? Well, I think no. We want to eat a good diet. It's one of the pleasures in life. But we did one study with obese people, mothers and children coming into Children's Hospital, and after taking our bar, they felt better. They all just loved the bar because they had felt much better than they'd felt in years. And so we gave them nutrition lectures and exercise and all of that. So I think this can be an incentive to teach people how a good diet makes you feel better. Anyway, we're busily working on it. And of course, there's no money to work on any of this stuff, but we'll get there. Now, life expectancy, as you all know, has been going up and up and up. Every decade, life expectancy goes up. And the top is women, the red dots, and the blue dots are men. And women live a little longer than men. And it keeps on going up. It's in our 80s now. And in fact, the US has just about the longest life expectancy if you correct for murders and car accidents. So we're doing better than Europe. But back in the 30s, when they set up Social Security, people lived about 60. They thought everybody'd be dead by 65. And every projection has been wrong since then. And the very last slide are the people who did all the work. Jung Soo, who's been working on ergothioneine, Joyce McCann, who's done all this theoretical stuff, Swapna Shenvi's working on immunology, David Killalay can analyze all the minerals at once, Harold Hellbach's a physician who's been working with David, Sandy Calloway's been working on mutation in mitochondrial DNA. We're collaborating with her. Mark Shiganaga is our obesity fellow. Michelle Meade-Snyder is a pediatric cardiologist, and she's been leading a lot of our clinical trials. And Ash Lau is a pediatric hematologist, and he's been helping on these clinical trials too. Thank you.